Okay, continuing on with the third example, this is a relative of the split plot. This is a repeated measures analysis. And before getting into it, let's just talk a little bit about the experiment itself. In this study, the main plot itself is going to have an underlying 2x2 factorial treatment structure, and the subplot, of course, is time. That's the whole point of the split plot, is that we are splitting in time. Specifically, dogs are our experimental units. And these dogs are randomly assigned to 4 different groups based on a 2x2 factorial treatment structure. These are the main plots. And so within each group, dogs either received morphine or trimethophan. These are some factors we are assigning to those dogs. And dogs had either depleted or intact histamine levels. So every main plot, every experimental unit or dog, is coming in one of those 4 flavors. And because we have 4 levels to our treatment, or a 2x2 underlying factorial treatment structure in 16 dogs, we know that there are 4 replications in this particular experiment. And then what we're measuring is the blood concentration of histamine at different time points after injection of the drug, so 0, 1, 3, and 5 minutes after injection. And because each dog, dog number 1 in the study is being measured at 0, at 1, at 3, and at 5, those 4 measurements on that dog are correlated to one another. In other words, they are not true independent measures of what the effect of time is, because they're being made on the exact same experimental unit, and that's the issue we're going to address here. So in this particular script, let's look at the data set first. I've loaded in the data, and I've referred to as hist.dat in the script. You'll see that we have dog as a column. We didn't talk anything about blocking in this particular experiment, so this dog is really your replication. You have dog 1, 2, 3, and 4 within each combination of your drug histamine time. So there's your 4 reps there. And we have drug, we have histamine. Of course, the combination of those two, the factorial combination of those two, is my main plot. And then we have the levels of time, which are the subplots. So these are all the factors involved in this particular study. When we load it in, we need, of course, to tell them that all those things are factors. And here we see that histamine is our response variable to number, and everybody else is a factor. So we should be good to go with a standard split plot analysis. So actually, let's do that first. Go down to the very bottom of this script, and you will see what this analysis would look like as a split plot. If we were to assume that these were not repeated measures in time on the same experimental unit, but that they were independent measurements of what the time effect really was, what this would look like is, because it's a CRD, it would be the main plot, which in this case is drug, histamine level, and drug times histamine level. These are the 3 degrees of freedom of the main plot, broken out according to its factorial treatment structure. We have the appropriate error term for the main plot, which we know in a split plot is rep times A. Well, rep is dog, and A is defined as this combination of drug and histamine level. So our error is rep times A, as it should be. And then, of course, we have B, which is time, and then we have A times B, and it's going to be all these different interactions. So this is our split plot analysis, and if we were to run this, let's just do that. If we were to run this, what we see is that our ANOVA table is broken up into 2 parts. We have a part dedicated to the main plot effect, in which the proper error term is rep times A, and we see all of that. And we have the bottom part of the ANOVA dedicated to the effect of time, or the subplot. And here we see a huge effect of time under an assumption of perfect independence, where in other words, we are saying that we have truly 3 degrees of freedom here for time, but we know that's not really the case. And if I bring your attention back to the decision tree here, we've started with a standard split plot analysis. We ran that, and we found that there was a significant effect of time. So our next step in this process is to run a conservative degree of freedom analysis. And that's what I do down in the appendix of the lab. We have the output of the standard split plot. And then down below, I have manually, so by hand, have gone through and said, well, I don't really have 3 degrees of freedom for time, I really only have 1 degree of freedom for time. In other words, I divided time by 3 to get 1, and anything that time is interacting with also gets divided by 3. So I adjust all my degrees of freedom. My f values all actually stay the same, but I recalculate p values here. And what I see is that I have agreement between the two approaches in terms of time being significant, but this drug times time, for example, here I'm calling it non-significant, whereas in the original analysis, let's look up here, drug times time was significant. So I have this lack of agreement between a full split plot analysis, a very liberal analysis, and a much more conservative analysis. And so this is what motivates going into a repeated measures analysis, in which we try to use the covariance structure itself to correct for the degrees of freedom in not such a severe way as a conservative degree of freedom correction will do. So at least for some effects in our study, we had significance under a standard split plot, but we lost significance under a conservative degree of freedom. And so that's what we're going to look at, this repeated measures approach using the easy ANOVA function within R. So going back to the script, to run that, you first of all have to install the easy library within R. This is not part of R's base package. So you're going to go over to your library, over to your packages, and install the package easy. I hope I have easy already installed here. I have already installed it, but I need to activate it for this session, so I'm calling on it. Once the easy library has been initialized, you can now call on functions within it, and this easy ANOVA is the function we're going to call up. And to use that within this particular example, the data set, of course, is hist.dat. My response variable is h. If you go back over here, h is the column for my response, and that's what it's asking for. The within ID, what this is asking for is actually the experimental unit as seen by the main plot. So my main plot treatments of drugs and histamine level, they were assigned at the dog level. So dog is my experimental unit ID. It is my rep ID. And of course, in order for me to be able to call upon it within easy ANOVA, dog has to exist within the data table itself, which it does. And we had to have made sure that R understood dog to be a factor, which is what line number 13 in our script was doing. So we designate the response variable, we designate the rep or the experimental unit for the main plot, which is dog. We then designate the within factor. This is the subplot, and of course this is time. This is the whole reason we're doing this. We're doing a split in time. So here's our repeated measures. Time, of course, to be called here, exists as a factor in the model. There's time. And then the between equals, what this is asking you for are the columns in the data frame or the data set that designate the levels of the main plot effect. Now our main plot effect is in fact a factorial combination of two different factors, drug and histamine level. So we can designate the main plot treatment by their interaction. And what that means is R is going to look at the hist.dat and is going to look at drug and histamine level, and the combination of those two, in this case M and depleted histamines, the combination of those two is the main plot treatment that was assigned to this particular dog. And then of course this was the histamine level measured at time 0 for that particular dog. If you go down the data set, here's that dog again on row 17. MD was the combination of the main plot level. It's still dog 1, and this was time measurement 1. So that 3.02 here and the 2.89, those are the repeated measures on that particular experimental unit. So that's the syntax for the easy ANOVA. You fill in everything and let's run it. So run that whole bit. Oh my gosh, we got all kinds of things here. Well, let's go up here. So it's getting mad at me here because my dog ID variable contains non-unique values, but that's fine. What it's saying is I talked about dog 1 through 4 for MD, and then I talked about dog 1 through 4 for MI, and I didn't use different ID variables. But R knows what's going on here because MD is different from MI, so this combination of MD1 and MI1, R knows that those are different experimental units, and so it's corrected that. It's automatically fixing that by generating unique ID labels. So that's great. What you'll see here at top is a first ANOVA table that is the exact results we got when we ran this as a split plot analysis. So let's just grab a value in here and take a look at it. For histamine, for example, we got a p-value of 1.75, 10 to the minus 13. Let's go look up here at histamine here. 1.75 times 10 to the minus 13. So when we manually pulled out the correct error term of A times rep and did our split plot, the Easy ANOVA is doing all that for us here. So this first part of the table is the regular split plot analysis with all the correct error terms. It's using the right error term for the main plot, and correctly it's using the residual error term for the subplot. Now, in this particular case, it's assuming complete independence of those repeated measures, which is not really true. So what we want to do is look at Mauchly's test for sphericity. It's running that test for us, and based on the covariance structure that it's finding within the data, it is suggesting that we deflate the degrees of freedom of time by a factor of 0.8. This is the Greenhouse-Geisser epsilon. So instead of using, let's go back up here, instead of saying time has 3 degrees of freedom, it's saying we should really use 3 times 0.8. That time really has about 2.4 degrees of freedom. And that's a little bit of a penalty we impose. And when we do that, what we'll notice is that for time, the p-value rises by about 2 orders of magnitude. And it's still highly significant. That's great, but we have been penalized a little bit due to the pattern of correlation in our data. In any case, that's about it. In this particular case, you would correctly, you would say that there is an effect of time. You would say that there was an effect of histamine level, an effect of drug. But of course, you would bypass all of those statements because what we're really seeing here is that the two-way interactions of drugs with time, the two-way interaction with histamine and time, and the three-way interaction are all highly significant. So what this is telling you is that you would want to proceed with a simple effects analysis in this particular case. And when you do that, you're going to be subsetting your data set by levels of time, and so the repeated measures disappears from that simple effects analysis anyway, and you get back to familiar territory. Okay, so that is running through the use of Easy ANOVA. I take a look at the lab handout. I've gone through a lot of narrative here. A lot of it's what I've gone over here, and then there's an appendix of what the full standard split plot analysis would be for this tortured dog example. And that's about it. Get in touch with me if you have any questions. Thanks.